Hello student, today we will start our topic heredity, in which we have earlier discussed about history of Mandel, terminology regarding to heredity and genetics. Now today we will discuss about hybridization. Mandel used pea plant to do his experiment on which he utilized that plant to do various kind of hybridization experiment. For in his first experiment, he took first a single characteristic feature and do the hybridization process for that. And such a kind of hybridization is known as monohybrid cross. So first of all, what is hybridization, and later on we will discuss about monohybrid cross. In our terminology segment, I have already told about hybridization. It's crossing between two plants for different features. Here you can analyze these features are basically alleles of a same trait. It's sexual reproduction which is going on between two plants for different alleles of same trait. That means if I am talking about height, suppose I am taking the example of plant height. In this case, I have to take two plants. I have to take two plants which are, the first one is tall and the one is dwarf. So these two plants, the first plant belong to capital T trait and another one should be small t small. It's called as pure tall and another one is pure short. So for the hybridization process, the first point is we have to take pure line if we want to follow Mendel's. Next one, the generation formed after the crossing of plants are called as filial generation. Here you can see progeny or offsprings are known as filial generation. Pure line always indicate the first pure line which we take, this one indicate to P generation or parental generation. While later on you can see various filial generations are there which are called as F1, first filial generation, F2, second filial generation, F3, third filial generation, and so on. Next one is about type of hybridization. On the basis of hybridization characteristic features, we can classify this one into various categories. Like first one is called as monohybrid cross. As the word reveals to you, mono, single, hybridization. It's a hybridization cross which is done for a single characteristic feature or single trait, you can write. As I told you earlier, example you can take plant height.
Plant color. Either the flowers are colored or colorless. You can take color is red and colorless as white. Just like that, another feature could be seed shape, whether they are round or wrinkled. So such a kind of characteristic features which are going to studied individually, then you can say that means if I am talking about plant height,
Rest of the characters feature are negligible. I'm not going to study them. If I'm taking only a single characteristic feature and concluding the result about them, then it's a monohybrid. The second is called as dihybrid. Di means two. If you are studying two traits together, then this is called as dihybrid process. I can give the example like this. You are going to study plant height along with flower color. That means I am going to study two traits simultaneously. In this one, tall and red is going to cross with short and white. Or you can take tall with white or short with red. But the parent should be pure in nature. This one is pure for first character and pure for another one. Same here. And just like that, this one is pure dominant for the first trait but pure recessive for the next one. But ultimately it's a pure characteristic feature. The third type of hybridization can be categorized as tri-hybrid categorization, tri-hybrid cross. Tri means three. If you are going to study three traits at a time, then you can say it's a tri-hybrid cross. In the example, I can write all the three characteristic features like height of the plant along with flower color along with traits, along with seed shape. So I should write the feature like this: capital T, capital T, capital R, and for this one I have to write another letter, that is round, crosses with small t, short plant, small r, white flower with white flower, and small w, that means wrinkled seed. And if I study more than three characteristic features, then this is included as polyhybrid cross. If we are taking more than three characteristics, this one can be categorized like this. The trait could be or you can say along with all these three features, I can take one more feature that is regarding to flower position. It could be axial or terminal, just like that. Up to the 10 syllables, there are two crosses only, mono and dihybrid, which I am going to discuss. The first one is monohybrid cross. Let us start with monohybrid cross. As I told you earlier, this one is a hybridization cross which is going on for a single trait. Here I am taking the trait which one was taken by Mendel, and that was regarding plant's height. He took two characteristic features. Plant height can be denoted as the dominant feature tall and recessive feature short. So he took two pure line parent which are tall, which is crosses with a short or a dwarf parent. Here capital T denotes to tall and small t for short plant or dwarf plant, and this one indicating to a P generation. That means parental generation. In the next step, they will form gametes. During gametogenesis, both the T get separate out from each other and ultimately we will get T kind of gamete. Capital T, you can see here. From the capital T parent or dominant parent, we will get capital T gamete. Gametes are always enclosed by a circle. In the case of small t parent, you will get small t kind of gamete. In the next step, fusion of gamete is done. As you know, these are the steps of sexual reproduction. The first one is gametogenesis and another one is fertilization. In the case of fertilization, this capital T kind of gamete get fused with the small t gamete and the product is capital T small t. Now as I told you in the terminology, if dominant and recessive both the traits are available, both the alleles are available, then only dominant can express itself, then the plant should be tall. Its phenotype is tall. Although in the case of genotype you can say or you can pronounce it as hybrid tall. This one stands for first filial generation, F1 generation. Now to form F2 generation, we have to do selfing of F1. So I am going to write here, it's selfing, in which capital T small t crosses with capital T small t. Then you can say by crossing in between F1 generation, you will find F2 generation. That means F1 crosses with F2 and you will get F1 crosses with F1 and you will get F2 generation. This one is the product of F1, which is a tall, 100% tall plant you will obtain here. And these are basically hybrid tall. And if you cross in between two hybrid tall, what you will get, that is about F2 generation. First, as I told you earlier, the first step is gametogenesis, in which these two factors get separate out from each other. So you will obtain two kind of gametes. These are capital T and small t. Now suppose this one is a male parent. Its pollen grain that contain the capital T character can fuse with another capital T and it can also fuse with another small t. Then the product will be either the combination can be expressed as capital T or it can be expressed as capital T small t in the next step. Just like that, this capital T can fuse with small t or this small t can fuse with another small t. Then the products are by the fusion of these two, the product will be capital T small t and the last product is small t small. So you can say this one is indicating to F2 generation. But now what is the actual geno and phenotype of these products? In the F2 generation, you can see some features are just like the parental generation, like capital T, this one is available here. Just like that, small t, it's also available here. And new combinations like F1. So right now I'm going to write about the pheno and genotype of F2 generation. First about phenotype. As I told you earlier, phenotype is the external expression of any characteristic feature which is written just before of you. This one is expressed as tall. This one is also a tall plant. Although this one is hybrid tall, it's also a tall plant and it's a short or dwarf plant. Tall, tall, tall and dwarf. They appear phenotypically tall. These three appear phenotypically tall and this one is dwarf. So these are expressed as tall and dwarf. Tall ratio dwarf. That is 3:1. 3 tall and 1 dwarf. While in the case of genotype, I have to express them like this. This one is pure tall, it's hybrid tall, this one is hybrid tall and this one is short. So the category can be categorized as tall, pure tall, hybrid tall and dwarf. Because dwarf is always expressed as pure dwarf. And the ratio is pure tall is capital T, which is only 1. Hybrid tall is capital T, small t, where both the alleles are available. This one is 2 in number and dwarf is small t, again this one is 1. So pheno and genotypic ratio for F2 generation and for monohybrid cross is 3:1 or and genotypic is 1:2:1. Just like this one, we will also discuss about dihybrid cross.